A very good morning to my dear students of level 3 semester 2, Faculty of Agriculture, Bangladesh Agriculture University. I would like to welcome you all to my part of principles of plant disease management. So, before going to the topic, I just want to remind you, why do we need to manage plant disease?
Sir, did the cluster record kora jeto?
If we don't manage plant disease. So, we need plant for our survival. So without plant,
Sir, যদি ক্লাসটা রেকর্ড করতেন ভালো হতো স্যার।
Okay, I'll do it later on, and I will provide you all the materials, both the PowerPoint slide and the other materials, so you don't have any problem, I think. So I am continuing the principles of plant disease management. We need plant for our survival. You know it. Without plant, the human being and the animal kingdom, the members of animal kingdom cannot survive, because we get food from the plant directly. There are a number of foods, for example, carbohydrate-rich food, protein-rich food, fat. We get all the essential nutrient elements, vitamins from the plant sources. And we are benefited by this, the function of this element. We also get oxygen from the plant. So the price of oxygen when we get sick, especially nowadays, the COVID-19 patient, they are to supply oxygens, synthetic oxygens, the cost of oxygens. So we are getting at the free of cost from the plant. So what happens actually, we are becoming benefited directly from the plant and also indirectly from the plant. So without oxygen, we cannot survive a single minute, because we take oxygen from the atmosphere, and with the help of oxygens, the glucose or the carbohydrate, this is actually react with the oxygens in the mitochondria of our cell and produces ATP. We called it energy and carbon dioxide. So we need ATP to survive. So how do we get ATP? We get ATP from the glucose and from the oxygens. And we get oxygen from the plant. We get glucose from the carbohydrate food, and that comes from the plant. So, the impact of plant in this way. But the bad news is that these plants are challenged by a number of threats, both biotic threats and abiotic threats. So we have to know about the threats. We have to know how to control those threats. We have to know how to manage. Actually, control is a term which is not possible in the nature in the true sense, but we can manage the disease to some extent. Okay. So our ultimate target of studying plant pathology is to manage the plant disease, and this management should be effective, economic, and eco-friendly at the same time. Okay. So you look here. So if we want to manage the plant disease, then we should have knowledge on different aspects of the plant, of the pathogen, and of course of the environment. The disease triangle. Look at this disease triangle. You can see here, there are three components of disease triangle. Number one, you can call host, then pathogen and environment. And there are interaction among these three components. Host interact with pathogen, pathogen interact with host, and both host and pathogen interact with environment. And it depends whether there will be disease or not. It depends on this interaction among these three components. For example, let's say rice is the host plant. For example, Bipolaris oryzae is the pathogen. Bipolaris oryzae causes brown spot disease in rice. So what is the role of environment in the disease development, in the brown spot disease of rice? Let's say if the environment is favorable for the host, for example, for the rice, but unfavorable for Bipolaris oryzae, then while the Bipolaris oryzae is present, but there will not be any disease. There will not be any brown spot disease because environment is unfavorable for the pathogen, for the Bipolaris oryzae. Okay. Let's say rice is the host plant and Bipolaris oryzae is the pathogen. So the environment is unfavorable for the rice and favorable for the Bipolaris oryzae. Only then there will be disease. Mind it. Let's say environment is favorable, rice is the host plant, but instead of Bipolaris oryzae, Bipolaris horochinina is present over there. Although environment is favorable for the pathogen Bipolaris horochinina, but and unfavorable for the rice, but still there will be no disease because Bipolaris horochinina is not the pathogen for rice. You understand? So there must be interaction among this pathogen, and whether there will be disease or not, it depends on the type of interaction. So if we want to manage the disease, what we have to do? We have to control either of these three components, or we can control host, we can control pathogen, or at the same time, we can control environment. Okay? Then we can manage the plant disease. Let's have a look what happens actually when the pathogen come in contact with the host plant. Think about fungi. What happens? So when pathogen come in contact in the host plant, then for successful infection, the resting spore of the fungus must germinate. After germination, they should produce germ tubes. In some cases, they produce appressorium. These are the specialized organ produced by the fungi before infection and also penetration phase. Thus, the resting spore germinates and produce these specialized structures. And you see, these specialized structures are produced outside the host plant. But when these pathogen enter the host plant with the help of germ tube or appressorium or penetration peg, then afterward they produce hostorium. In case of the biotrop, and after entering, the fungus produces toxin, especially the necrotrop pathogen produces toxin inside the plant. They also detoxify the chemicals that resistant to the plant. For example, phytoalexins. These phytoalexins produced by the plant, and these phytoalexin tends to kill the fungus. And what the fungus does? In this case, fungus detoxify the phytoalexins, and afterward, after detoxification of the phytoalexins, they go for reproduction. Their growth, development, and reproduction. That is, they increase in number. So the host plant, for example, will not be silent when they get infected with the pathogen. They will not be silent anymore. You understand? So there will be also response from the host side. there are a number of responses. You can see here, the host plant will produce papillae over here. Outside the host plant, on the surface of the host plant, they will form papillae. This inhibit the entry of the pathogen inside the plant. And afterward, inside the host plant, the plant produces cork and lignin layer. This makes the leaf tissue, for example, or the stem surface thick, hard. As a result, the pathogen will not be easily able to enter the plant. And the plant also produces some chemicals. Their signal system become activated as osmotic acid signaling pathway, salicylic acid signaling pathway. Those pathway will be activated. For example, although the pathogen come in contact with the leaf on the surface of the plant, but all other plant part will be alert that they have been infected. As a result, they will produce some chemicals. The chemicals responsible to inhibit growth of the pathogen, development and reproduction of the pathogen. And sometimes those chemicals kill the pathogens. You see phytoalexin. So sometimes when the plant recognize the pathogen, the plant protein, when recognize the pathogen protein, then there will be hypersensitivity. That is the localized death of tissues. So as a result, those infective propagule cannot disseminate, cannot spread over the plant tissues. So local, those tissues will be dead. We call it hypersensitivity. So this is a kind of resistance reaction, mind it. So when we see that there are some dead tissue in the plant, very small localized dead tissues in the plant. So we have to think that the plant is showing resistant reaction against the pathogens. So this is not only the death of the tissues, but this is the resistance of the plant. In this way, the plant prevents the dissemination of the pathogen, spread of the pathogen from the area of presence to the other healthy areas. Okay, so this is what is happening when the plant come in contact with the pathogen or pathogen enter the plant. Then we have to think about how we can manage the disease.
So could you please repeat the previous line?
Yes. I will provide you the slides, so you don't need to worry about this. Okay. So we have to think about how we can manage the plant disease actually. When the plant infected with the pathogen, how we can manage. There are mainly two systems, immunization and prophylaxis. The cure of diseased plant is not possible because the disease becomes visible only after injury to plant has taken place. So when we see that the plant is diseased, already the damage is occurred inside the plant. We see the symptoms after infection, not before infection. When we see symptom, the plant is already damaged to a certain extent. What we have to do? We have to prevent the pathogen so that there is no disease. Mind it. So unlike the human being or animal being, the diseased plant can never be transformed to a healthy plant. For example, when we become sick after medication, we become healthy again. But in case of plant, if the plant is diseased, that diseased plant or that diseased tissues will never come to healthy tissues anymore, even after application of pesticide or combination of all other techniques of disease management. that diseased tissues will never return to healthy tissues. But what we can do, we can protect the remaining healthy tissues, or we can protect the remaining healthy plant in the field by following a number of methods, chemical, biology, and other methods that Professor Atikur Rahman Khokon has discussed. Okay. So what we have to do, we have to take some preventive measures, and we can do it by using the seed of resistant variety or by cultivating the resistant plant. And let's say we have used the resistant variety, the seed of resistant variety of resistant plant, but still there is a chance of disease in the field because the resistant plant is not resistant all over the time, all over the area. The resistant plant that is resistant today, it will be susceptible after 2 years, for example. The resistant plant that is resistant in Bangladesh may be susceptible in India. Okay, so in this case, the resistant plant might also be diseased, and we have to take some measures. We call it protection measures by prophylactic approaches. Let's have a look. What happens actually if we want to control or manage the plant disease? You see here, there are mainly two approaches, immunization and prophylaxis. And immunization confer the resistance, and the resistance we can, in some cases, we can induce by applying some inducer. We call it chemotherapy, especially those are the chemicals, for example, salicylic acid, jasmonic acid, and other resistance inducer hormones. Okay, and what is prophylaxis? It consists of three principles. You can see here: protection, that is after infection of the plant, we have to take care of the plant. Then eradication, that is complete destruction of the disease-causing element from the plant or from the soil or from the environment. So eradication can be done by a number of methods. You see here crop rotation. So if we cultivate the same crop in the same field years after years, then the incidence and severity of the disease will be increasing with the passage of time. So we have to rotate the host plant. In this way, we can actually eradicate the pathogen. Not fully, but some extent. It takes time. It takes time for 5 to 10 years. The pathogen can survive in the plant debris. After harvesting, there are still some straw. There are still some plant part remaining in the field after harvesting, and pathogen will be present with those remaining plant part. Then what we have to do? We have to clean the field after harvesting. We call it sanitation. And of course, we can apply chemical pesticides to kill the pathogen. And we have to destroy the host, alternate host and collateral host. You know, the alternate host and collateral host. So after harvesting the main crop, the pathogens survive in the alternate host. For example, Bipolaris oryzae can survive in Durba grass. So Durba grass is the alternate host of Bipolaris oryzae. So after harvesting the rice, we also have to destroy the wheat, that is Durba. And we can do it by burning the plant, by sanitation, by applying chemicals, and by crop rotation as well. Then another approach you can see here, the legislation, that is deal with the law, enforcement of law and regulation of the pathogen. How can we do it? Let's say we are fear about the coronavirus. So this virus was first reported in China, you know it. So how? So what is legislation in this case? So we have to follow quarantine. That is, we have to check the coronavirus in the airport, in the stall port, or in all other checkpoint of in the border area. So we have to check whether coronavirus is present, is not. If coronavirus is present in the people moving to Bangladesh, coming to Bangladesh, then we have to restrict him or her. So this is called quarantine, and we can do it by state-wide or by centrally. So there is agency in the world which control these things, and by doing so, we can prevent the entry of pathogen. For example, lead blight of potato was first reported in the Europe. But how this pathogen, Phytophthora infestans, present in Bangladesh? Somehow, with the infected tuber, the potato, with the potato infected potato tuber, we imported the infected potato tuber, and with the potato tuber, Phytophthora infestans causing lead blight of potato, it entered Bangladesh somehow. So if we could prevent this, then there will be no lead blight disease. So this is called legislation with the help of quarantine. You know, there are a number of quarantine stations in the airport, stall port, and seaport. So there is quarantine officer, the people like you after graduation, after BCS, some of you might be assigned as quarantine officer in the airport or stall port or seaport. So your function will be to check the imported materials for the presence of pathogen that is not present in Bangladesh. Okay? So we have to prevent the introduction of those pathogens from outside our country. This is called legislative measures.
Sir, state and central difference.
So state, in this case, this is, for example, in case of USA, you know, there are 50 states, and there is a governor of each state and central government, federal government, not applicable in Bangladesh, but in case of USA. But in the sense that the world has an organization that regulates the quarantine technique, quarantine, and each country regulate the quarantine in their own airport or stall port or seaport.
Sir, collateral host or alternate host কি একই জিনিস?
So you know it from level 2 semester 2, the difference between collateral host, main host, and alternate host. So alternate host, you see here, the pathogen can survive in absence of the main host, in the collateral, in the alternate host. Okay, this is the pathogen can complete the life cycle and in presence, in absence of the main host, in the alternate host. But collateral host is not important for the pathogen to complete the life cycle. The pathogen cannot complete the life cycle. They just can survive. Okay, you can check it from level 2 semester 2.
Sir, can anybody bring any seeds of crop from other countries like Australia, as they check the disease, whether there is any disease or not? So is it permissible to anybody?
So your question is, can you repeat your question?
So I'm saying that can anybody take any seed from other countries?
Okay, of course, we can. So let's say when you are coming back to Bangladesh from Australia, you want to, when you land at Dhaka airport, for example, or the Zahangir International Airport, then you have to report to the airport authority that I am coming with some seeds from Australia. Then the airport authority will check. The airport authority means the quarantine officer of Zahangir International Airport will check your seed, whether any pathogen that is not present in Bangladesh is present inside your seed or not. he or she will test the health of your seed. We called it seed health testing. You will learn it in level 4 semester 1. So there are some methods and techniques, some advanced method, molecular based, PCR based method. Within 2 to 3 hours, the quarantine officer can check whether your seed contain any pathogen that is not present in Bangladesh. So after testing, you will issue a certificate. So if the pathogen is present or not. If pathogen is present in your seed lot, then you are not allowed to take the seed outside the airport. So those seed will be damaged or destroyed immediately. So once they confirm that the quarantine pathogen is present within your seed. You got it? If there is no pathogen, for example, then he will issue a certificate that your seed lot is free of any quarantine pathogen, and you will be allowed to take the seed outside the airport.
Sir, is it possible to check all the seats?
No, it's not possible to check all the seats. So what does the quarantine officer do? The quarantine officer will take sample from your seed lot. For example, you are importing 100 ton of seed, for example, rice seed or potato seed or wheat seeds from China or from Australia or from Europe or some other countries. Then what will the quarantine officer do? Then quarantine officers will take sample from the seed lot. Sample means the representative portion from the lot. So after taking the sample, they will take the composite sample from each packet. He will take the sample, then he will mix the sample, all the sample. After mixing, we get the composite sample, and from the composite sample, the quarantine officer will collect the working sample, just 1 or 2 grams. Okay, and the accuracy of the certificate completely depends on the accuracy of sampling. So the sample must be representative. You should not be biased. Okay, so during sampling, you should not be biased. You must follow the methods of sampling so that your sample is representative. And from the working sample, from only 1 or 2 gram seed, the quarantine officer will check, and the sample number, working sample number, will vary depending on the amount of materials you are importing. So if you are importing, for example, 100 ton of seeds, then the working sample might be, for example, 100. Okay, so after getting the result, they will make average. For example, in one seed lot, there is no, in one working sample, there is no pathogen. For example, out of 100, in 70 sample, you did not get any pathogen. But the rest of the 35 samples, you got pathogens. So you have to make it average. So what is the number of pathogen? What is the severity of the pathogen in the imported seed lot? And there is standard limit for each crop. Okay, I will discuss it later on. You can check here. So I have already discussed these 3 components of disease triangle. So we have to know the host, we have to know the pathogen, we have to know the environment. So I have 10 more minutes, so I have to go fast. So you know that we want to manage the disease. So what we have to do? So we have to look for the resistant variety. And we can make the plant resistant by a number of ways. You can see here by diverse sources of resistance. There are several resistant varieties might be available of the same crop. So we have to cultivate not only the one resistant variety of the same crop, but a number of resistant varieties if available, we have to cultivate in the field. Then we can pyramid the genes for resistance, that is accumulation of several resistant genes in the same plant. We can deploy genes for resistance in space and time. That is, the resistant variety one variety is resistant, for example, in Mymensingh, but may not be resistant in Khulna. Okay, the variety might be resistant in 2020, but may not be resistant in 2022. Okay, we have to consider all these things, and we can make the plant resistant by applying some pesticide, by applying some resistance inducer that can make the plant resistant. And how we can control the pathogen? Can we not convert virulent pathogen to avirulent pathogens? So what we can do, we can exclude the pathogen, or we can reduce the number of inoculum in the infected plant part or in the infected areas. And we can do it by quarantine, you can see, by seed certification scheme. I have already discussed. Okay. And we can apply the chemicals, pesticide, to kill the pathogen. Okay. By doing so, we can reduce the initial inoculum, or we can reduce the infection rate. Okay. So elimination of existing pathogen, we can do it by a number of chemical pesticides, or by following different physical methods that Professor Atikur Rahman discussed. And we can eliminate the pathogen by crop rotation, I have discussed. And we can reduce the initial amount of inoculum by seed treatment and soil treatment, because the pathogen present in the seed. So we have to treat the seed, we have to treat the soil in order to kill the pathogen present in the seed and in the soil. And we can do it by a number of chemical, for example, Vitavex, for seed treatment by formalin or formaldehyde. In case of soil treatment, we can dip the fruit in the thiobendazole solution. Okay, and of course, quarantine practice and preventive treatments will help us to control the pathogen, to eradicate the pathogen, and to reduce the number of inoculum in the infected plant or in the infected areas. Okay, I have already discussed, we have to spray the chemical, either protective or systemic chemicals, to kill the pathogen. Okay, so what happens actually, we don't apply chemical to control a particular disease of a particular crop. For example, we only apply chemical to control a devastating disease of a very important crop. Okay. For example, there are some major crop, there are some minor crop. Rice is the major crop in Bangladesh, and barley, this is not a major crop in Bangladesh. So we'll not apply chemical to control the pathogen of barley in our field. It depends on the importance of the disease. It depends on the importance of the crop being cultivated in a particular area. Then we have to modify the environment. We cannot control the environment, we cannot control the temperature, relative humidity, but we can modify the temperature and relative humidity to some extent. And by following a number of approaches or techniques. For example, by adjusting the planting time or sowing time of the seed, by reducing the density of the plant, by changing the irrigation pattern, by changing the timing of irrigation. By doing so, we can regulate the temperature, relative humidity, and other aspects of environment. You can see here, we can regulate the temperature by adjusting the sowing time to avoid burnt and flag smart of wheat. And we can use the solar heat to eradicate internally seed-borne pathogen of loose smart disease of wheat and barley. We can treat the seed by hot water to eradicate black rot bacterium from cauliflower seeds. But these are just some examples. There are numerous examples. You can check it. So by doing so, by adjusting the sowing time, we can actually modify the environment. And we can regulate the moisture by providing irrigation. Potato scab disease is very severe in the dry condition. But if we provide irrigation in the potato field, for example, then the severity of the scab disease afterward will be less. Then flooding of soil can be effective in eradicating fusarium wilt. So fusarium fungus cannot survive in excess amount of moisture. Okay, and there are some pathogens which are abundant in alkaline soil. For example, sclerotinia. So these fungus are abundant in the alkaline soil. So we have to make the soil acidic by lowering the soil pH by applying sulfur fertilizer. And we can manage the nutrient status. There are some pathogens which like nitrogen, there are some pathogens which like potassium, there are some pathogens which like phosphorus. So we have to know the nature of the pathogen, and by doing so, we have to regulate the pathogen by manipulating those nutrient content. And we can follow some cultural practice that I have already discussed: sanitation, the tillage, then deep plowing, spacing. That is, some disease spread from one plant to another plant very easily if the plant spacing is minimum. So we have to ensure adequate spacing so that the pathogen cannot spread from the diseased plant to the surrounding healthy plant. There are some examples, you can check it. This is one of the factors in the disease triangle or tetrahedron, but we cannot do regulate this time dimension all the time. But what we can do, we can just adjust the sowing time, or we can select some early maturing variety. By this, we can avoid some disease. So there are actually 4 principles of plant disease management: exclusion, eradication, protection, and resistance. Initially proposed by Wetzel in 1929, and later on other principles added, for example, avoidance and therapy. What is avoidance? You can see here the methods and practice which avoid the contact of the host with pathogen or susceptible stage of the plant and conditions available for the pathogen should not be coincide. So by ensuring this, we can avoid the pathogen, and we can do it by a number of ways: by selecting the area where pathogen is not present, by selecting the field, by choice of time of sowing. For example, lead blight is very severe in the month of January and December, when temperature is very low, relative humidity is very high. So we have to sow the potato tuber in such a time so that we can harvest the potato before January or before the environment favorable for the phytophthora infestations. So by this, so we can avoid the lead blight disease of potato. We can select some disease-resistant varieties. We can modify the environment and so on.
Sir, time share, sir.
Okay, I will finish it. And I will continue with rest of the principles in the next class. So the next class will be on Monday, right?
Yes, sir.
Okay. So I have provided the link for attendance. I can check that there are 295 students present in the class at the moment. So please provide your attendance to me to my email address, or I'll check it in the Google. Okay. Any question?
স্যার বুঝলাম না স্যার অ্যাটেন্ডেন্সের ব্যাপারটা।
So I have provided the link, Google form. Did you get it? I have sent it to the CR of section C.
জি স্যার।
Okay. So there are 295 students present. So please give me your presence over there.
Okay, sir.
Okay, thank you for your patience. See you on next Monday. Any question?
না স্যার।
Thank you, sir.
Thank you so much, sir.
অনেক ধন্যবাদ স্যার। আসসালামু আলাইকুম।
Rabiur Rahman.